All right, so let's get right into it. In the world of poultry farming, there's this really persistent and costly mystery that's been bugging chicken flocks for years. It's a virus that, no matter what we do, just keeps coming back, but in some pretty surprising new forms. So today, we're going to unpack the wild genetic detective story of how scientists track down a viral shapeshifter.
And look, the stakes here are incredibly high. We're not talking about some minor bug. This thing, the avian infectious bronchitis virus, or AIBV, costs the U.S. economy millions of dollars every single year. That's a massive hit from lost production and sick birds, which is exactly why getting to the bottom of this mystery is so important.
So yeah, let's properly introduce the villain of our story. This is all about a super contagious and relentless coronavirus that basically has the entire poultry industry on high alert all the time.
AIBV is, to put it mildly, a huge headache. It's a type of coronavirus that just hammers chickens, causing really nasty respiratory and kidney disease. Now you're probably thinking, well, just vaccinate them, right? And we do. Mass vaccination is standard practice. But here's the real puzzle. Even with all these vaccinations, outbreaks keep happening, and sometimes they're really, really bad. So what gives? Why isn't the vaccine doing its job?
That is the exact question a team of scientists wanted to answer. So they basically put on their detective hats and started hunting for clues right there in the infected flocks, trying to track down the specific viral culprits that were causing all this trouble.
Okay, so first things first, the investigators had to figure out where the problem was the worst. So they measured what's called the viral load, which is really just a fancy way of saying they counted how much virus was actually present in different flocks. And as you can see from this chart, a couple of these bars, flock 4 and flock 6, are way higher than the others. Bingo! These were the hot zones, the perfect place to start digging.
And that's how they got their two prime suspects. From that flock with all the respiratory problems, they pulled out a strain they named AIBV4. Then, from the other flock that was having kidney issues, they isolated AIBV6. Now, the fact that these viruses were found in totally different parts of the body, well, that was the first big clue that even if they looked similar, they might be behaving very differently.
So, with their suspects in hand, it was time to head back to the lab. The next move was to do a kind of genetic fingerprinting, to read their entire genetic code and find out who these guys really were. They do this using a technique called genome sequencing. Honestly, the best way to think about it is that scientists can literally read a virus's entire instruction manual, page by page, letter by letter. It's how they can see exactly what makes a virus tick and, who it's related to in the viral world. And this is what they came up with. I know, it looks a little complicated, but just think of it like a viral family tree. The closer two branches are, the more closely related the viruses. So when the scientists sequenced their two suspects, AIBV4 and AIBV6, and popped them onto this tree, they found something that at first made absolutely no sense. It turns out that these new, dangerous viruses weren't some brand new invader from far away. No, they were incredibly close relatives of MAS41, which just happens to be one of the most common vaccine strains used to protect the chickens. The bad guy looked almost identical to the good guy. And that was the first massive plot twist in this whole investigation. So even though these new strains looked like the vaccine, they sure weren't acting like it. The scientists found a very specific genetic signature, this tiny piece of code right on the virus's spike protein. Think of it like a serial number on a weapon. This specific sequence was a dead giveaway. It was direct proof that these viruses were built to be dangerous, not harmless. And that brings us to the absolute heart of this mystery, the discovery that just flipped the whole case on its head. It's a huge plot twist that explains how a protector could suddenly be linked to a killer. So, this was the million-dollar question, right? If these new viruses are practically cousins with the vaccine, why in the world are they making chickens so incredibly sick? Well, the answer lies in this fascinating and honestly kind of terrifying viral superpower. And that superpower is called viral recombination. Okay, so get this. It's a process where a virus can literally swap bits and pieces of its genetic code with other viruses that just happen to be in the same cell. It's like a genetic mashup. The result? A brand new hybrid virus that's part old, part new. It's like a genetic disguise that lets it evolve at a crazy fast speed. And right here, this is the smoking gun. These charts are mapping the entire genetic code of our two suspects. See all those different colored blocks?
Each one represents a chunk of DNA that was clearly borrowed from a totally different parent virus. These aren't pure strains. They're genetic patchworks. They're basically Frankenviruses, stitched together from the parts of many different parents.
And then came the final jaw-dropping revelation. The analysis showed that one of the main genetic donors was the vaccine strain itself. The live weakened vaccine virus, the one designed to protect the chickens, was in there swapping genetic material with wild viruses. And by doing that, it was accidentally helping to create new, dangerous strains that could evade the vaccine. The protector had become an accomplice.
So what in the world does this all mean for the future? Well, this investigation paints a much bigger picture about this constant, high-stakes dance between us and viruses and how they evolved in response to everything we throw at them. Okay, so let's just quickly recap this whole genetic detective story. We learned that viruses use this trick, recombination, as a super powerful tool to evolve. We saw, pretty shockingly, how this process can mix genes from both wild viruses and the vaccines we use, creating totally unpredictable new threats. This means we need what the paper calls vigilant monitoring, which is basically just constant surveillance, watching these viruses change in real time. And of course, it means the next generation of vaccines has to be way smarter, designed to outwit an enemy that is constantly changing its disguise. And really, this whole case leaves us with a much bigger question. It just goes to show that we're in this perpetual arms race with the microbial world, right? Every time we build a new defense, they find a new way to get around it. So the ultimate challenge is, how do we innovate faster than evolution itself? How do we stay one step ahead of a constantly changing enemy?